Okay. Hi everyone, thanks for coming to this month's Toolbox. It's our second last Toolbox for the year. And today we have Dr. Natasha Karenyi speaking. She's a marine benthic ecologist and also lecturer in the biology department, as well as a core SEEC member. Her work focuses on consolidated sediment habitats, and she uses novel stats to analyze the data for this. Today she'll be speaking about mvabund. Thanks, Natasha.
Thanks, Bianca. Okay, we just do a sound check, guys. Welcome to the guys in the Adobe Connect room. If you can hear me, just write that in the comment section and question section. Hi everyone, thank you for coming. This journey kind of started a few years ago, where I've always worked on multivariate analysis, but more recently, probably in the last 10 years, there have been some new developments in terms of model-based multivariate analysis, and this is one of the packages that I'm looking at. But before we start, happy Halloween! To those of you that celebrate, have a good one. So when do we use multivariate analyses? Essentially, when you have multiple response variables, right? So that can be in ecological data. There are many different types of data that would fit that description. We often collect species-related data in surveys or experiments. So you've got a species, and then a species list with an abundance at each site that you sample. So abundance is kind of a broad term. It can mean many things. So we include things like presence, absence, ordinal, count data, biomass, and percentage cover. Then in terms of environmental variables, usually relating those to the species, we would have geological data, oceanographic or climate-related data. You can also have morphological or traits data that would be multivariate, so size and shape measurements, life history traits of organisms, and so on. And also molecular data makes use of multivariate analysis quite frequently. So there are two broad types of analyses. There's the association-based analyses, so that would be your correspondence analysis, your primer analysis, multidimensional scaling, those kinds of things, cluster analyses, and then there are model-based ones. If you're interested in more information about the association-based ones, we did a SEEC toolbox about exploratory methods a few years ago, and so that's available on the SEEC website. And so now we're going to focus on the model-based ones, but also you can also do exploratory analysis with model-based multivariate analysis, but we're not going to talk about that today. If you look at that introduction to multivariate analysis, I do mention some of the types you can use. So today we're going to talk about the mvabund package. It was developed by Professor David Walton. He's based in Sydney at the University of New South Wales. And he visited us last year and did a course trying to introduce this analysis or these analytical methods to us. And it's not used for exploratory analysis. It is used for statistical inference. And there's a definition of what that means. But essentially, instead of saying something only about your specific area that you sampled or your specific samples, you want to say something about the effect of a treatment or of environmental variables across a much broader scale, and to do that you use statistical inference. For example, if you're looking at the impact of mining, you don't just want to look at the impact of mining in your little area that you're working on. You want to say something generally about the impact of mining at a broader scale. And this is very dependent on your sampling design. So if you are going to design your sampling strategy, you should probably look at this other SEEC toolbox that we did on experimental and survey design, which would be really useful in that sense. So The mvabund package is still being developed, new methods and new ways of analyzing the data are being developed every year. So currently, it can be used on all these different data types except for percentage cover. And hopefully they will include that in future. So the characteristics of the multivariate data that are really important are, one, that they're multivariate, of course. So they have many correlated response variables, and often these variables, there are more variables than observations. For example, you can have 20 sites and 100 species. So variables here are your species, and observations would be sites. And also that you're dealing with abundance data. So abundance or presence-absence data usually has a very strong mean-variance relationship. So as your numbers, as your mean gets higher, your variance gets larger. And those are the two characteristics that mvabund is trying to incorporate into the analysis. Okay, so the function that we're going to talk about mostly today is the mini-GLM function, which is designed for this kind of abundance data, and it deals with those two properties that are really important. So in terms of the multivariate aspect, it uses this row, which is site resampling, in order to use that for inference, and also to preserve the correlation between the variables. So you want to make sure that the relationship between the species is maintained, is intact. And then the other aspect is the abundance, where the mini GLM actually fits separate GLMs to each species. And then you can use family in order to define that mean variance relationship, and you can use plot to check your assumptions related to these models. Okay, so what we normally want to know is, for example, does a treatment have an effect on an assemblage? So that's where you're using discrete explanatory variables, right? So you have a treatment, you have a block, you have a year that you're sampling across different years. That would so that would be your discrete explanatory variables. And then following that, you want to know what are the indicator species. In which species is that effect seen, essentially. So the data required for your mini GLM. So if you look in the actual mvabund package, you'll find all the data sets are lists with 2 or more data frames. One would be your species abundance data per site, and then another type would either be your treatment or your environmental data per site. You can bring these in as matrices and then convert them when you're in mvabund if you're using your own data. But for the purposes of today, we're using data sets from mvabund, and most of the code is actually found in the reference manual, although I've shuffled things around to make it a little bit more coherent. So the first thing that they do is they convert abundance of your species to an mvabund object. This is the mini GLM only works if you input an mvabund object. So that changes the structure of the data so that it suits this particular set of models. And then the second data set would be your treatment or your environmental variables, which are either there as a vector in the case of the factor like a treatment, or a data frame if you're using environmental data. So just by way of an example, so this is one of the data sets from mvabund. In Tasmania, they looked at benthic copepods, and they had 12 different copepod species. Their sampling design was they had these different blocks. They're not necessarily next to each other like this, but different sites that they sampled, and in each of those areas, they had plots that were disturbed and undisturbed. And you can see this is a very nice, neat, balanced design. And so once you bring in the data, we want to visualize it. The first thing you have to do is you have to create your mvabund object. Okay? And this is what the object looks like. So you have, instead of now species, instead of, whether it's which block it's in and what disturbance level it is, you just have 1 to 16. Those are your observations. And then you have your species, and you have to have everything in a specific order. So they'll have block 1, disturbed, undisturbed, block 2, disturbed, undisturbed, and so on. Okay, so that's the general layout of an mvabund object. And then you pull in your treatment, which is a vector, and your block, which is also a vector. And then you can plot these in relation to each other. So you can say your mvabund object, so you plot your mvabund object and as a function of treatment, and then you can color it as numeric to the block. And this is a really nice way to visualize your raw data. And then it disappears. Hold on a second. Yeah. Sorry guys, I'm just gonna run it off the computer on the that other one. Okay, should work now. Okay, perfect. Sorry. Technology, right? Okay. So this is the way you visualize the data. It'll have your species. It does a maximum of 12. The 12 most abundant species, essentially, is what you can visualize. So they'll have your species and then some measure of abundance on usually a log scale. And then because we told it to color it by block and we gave it the treatment as the predictor variable, it now separates for each species the abundance in the disturbed and the undisturbed system plots, as well as the block that it was in. So it's color coded to the block. So it's a really nice way to look at your data, but again, it's only for the 12 top species. So and in most of our data sets, we would have many more species than that. But it is a useful tool. Alright, so then when you specify your GLM, you have to specify 3 general components. Your random part, which is your probability distribution for your response variable. So that defines your mean variance relationship. Right? Then the systematic part, which is your explanatory variables and how they relate to, and kind of the form of those explanatory variables. And then you have a link function that links that random and systematic part together. And so this is common for all GLMs, and it's no different for the mean GLM function. It does have a few assumptions, same as GLMs in general. So observed Y values are independent after conditioning on X. Y values come from a known distribution with a known mean variance relationship. So because we specify that, that's usually met. There's a straight line relationship between some known function and of the mean of Y and each X value, or each X, and also that the residuals have a constant correlation matrix across observations. You can change all of these things. You can use different arguments to better specify for your specific issue or for your specific problem you're trying to answer. Okay, so because we're using count data, Generally, there are two different mean variance relationships or probability distributions that you can use for count data. The Poisson distribution, which assumes the mean a 1:1 ratio, the mean variance relationship, and then a negative binomial, which usually is the probability distribution you go to when Poisson doesn't work, it's over dispersed. So basically, like I said, when you specify your mini GLM, you've got just what we're calling it. Say mini GLM, you put in your function. So it's my mvabund object, and we think block and treatment are probably going to be but both are probably going to be important, and we're wondering about whether there's an interaction between both of them. It's the multiplication sign here. And so we're going to try with the negative binomial, and we're going to try with the Poisson, and we're going to see which actually fits better. Right. So now we can plot the residuals for those once we've run them. And with the Poisson, you can see that there's this funnel shape, which means that it's over dispersed. So now, the negative binomial actually looks a lot better. So we're going to go with that model. Right. You can also check for normality if you need to. Okay, so then the workhorse of this particular program is actually the ANOVA. This is where you get all your results out. And so this is an analysis of variance table for our model that we specified. And you've got your block treatment and your block treatment interaction. And you can see that block is definitely significant, as a significant effect, so does treatment, but there's no interaction between them. So essentially you're getting the same directional response in treatment for all the different blocks. So you don't have to worry about that. Okay, so that's great, there is an effect. Now we need to figure out in which species is there an effect, right? So again, you can use the same function, so ANOVA. Say all of this is the same, but now we're going to add this argument called p-uni. And p-uni is where you do univariate tests using the same equation for all the different, for every single species. Okay. And we chose adjusted because we want to control for the family type 1 error. So we just want to make sure that when we're doing multiple testing, we don't exceed our 0.05 error, 0.05 alpha for the testing. Okay, so then essentially what you do is you get all the same information here, but then you get the univariate tests at the end of it. And so for each species, for each of the 12 species, there was a result, but I've just taken the first 3. And you can see that in terms of treatment, there's a significant change in your myriad species. And so that would be what you call an indicator species. But if we look at the abundances again, so now we're looking just at this line with the myriad species, you can see that the disturbed sites always have lower abundance than the undisturbed sites, and within each block, that's true. So here's the green one, there's the green undisturbed, and there's the green undisturbed. There's the blue disturbed and the blue undisturbed. So it's nice to be able to look at the actual abundances and then to be able to say, actually, yes, there are these differences in them. Right, so now we know the treatment had an effect, and now we know that what the indicator species are. So now the next question that another type of question that we normally ask is, which environmental variables are most strongly associated with an assemblage. And here we have continuous explanatory variables, more often than not. And so we're going to use a different set of data, also from mvabund. Here we've got species abundance, again, count data for 12 spider species, 28 sites that were sampled, and you've got 6 environmental variables that were measured at the same time. So generally you would check for collinearity. Some things maybe, nothing pops out too strongly. And so again, you're going to do your SPI-DAT, which is your mvabund object, where you take all your abundance data, you restructure it into the mvabund object. And here we're going to use a data frame because you're using more than one environmental variable. So you define your environmental variables, data frame. Okay, then again, you specify your mini GLM. You're using SPI-DAT, and it's related to your all your environmental variables in the data. You'll note that I didn't specify family in this one because in the mvabund your default is actually negative binomial. So if you're using another type, Poisson or negative or normal binomial, then you'd have to specify it. But negative binomial is the default here. Right, so now if you're looking at conditional effect, so that's what predictors add the most to the deviance with when all the other predictors have been or have already been fitted, then you will get, you can see here that the soil dryness, herd layer, and reflection are the ones that come out most strongly as the most significant contributors when everything else has already been fitted. But now the other option is to look at marginal effects, and there isn't a very easy, elegant way to do that in mvabund yet. So when you consider the marginal effects, you have to calculate it for each of your environmental variables separately. But essentially what you come up with is that bare sand now is very important and moss is very important. And when we look at the data, then you can see that in terms of dry soil and bare sand, there is actually a relationship. It's not a linear relationship, but there's a threshold beyond which you have bare sand and you don't. And moss also seems to have quite a strong relationship with soil dry, so maybe that's masking it, essentially. So it's always good to interrogate the data after you've had a look at the results. Okay, so now what do we want to know? We know we can determine whether treatment has had an effect, what the indicator species are, and which environmental variables are most strongly associated with an assemblage. These are the basic questions that we would ask in terms of an ecological question. So, as I said, the mini GLM can handle some of the most common families that we need in ecology. So, negative binomial, that's for over-dispersed counts, the log-link function, the Poisson, the binomial. There are two different ways to deal with presence-absence data. They've got these two different link functions, but essentially that's as much as the mini GLM function can handle. And so they introduced another function called the mini any function, and this function is used mainly for compositional change. So it works on relative abundance. It's more flexible than the mini GLM. And you can use many different models with it, not just the GLM, so you can use GAMs or others. And then it adds another family to the analysis so that you can deal with things like biomass or more obscure measures of abundance. I've seen them use it with, on a coral reef, they used the number of centimeters in a 10-meter transect that touches a particular species. So there are different measures that you can use of measures of abundance. You can use using this many in function. And then they also have a function called the trait GLM. So this is kind of their response to the whole idea of the fourth corner analysis, where you're trying to understand how the traits of your organisms are related to the environment. And normally what we would do is we would take the environment variables by site, the species by site, and the traits by species, and you would multiply the matrices of the traits and the abundance together to try and then relate that to the environment on a site basis. But in this function, they're turning it around and they're trying to predict the abundance of species based on your environmental variables as well as your traits variables. And so this is kind of a new, different take on how to deal with the function, with understanding how traits are related to your environment and to the abundance of your organisms. And that went really faster than I thought it was going to go. But yeah, so there's lots of information. I've got a few here, some of the most important papers, some websites, blogs that you can have a look at that would be really interesting. Some information about this particular distance-based, the comparison between the distance-based and the model-based ways of analyzing your data. Yeah, and I mean it's really a new kind of system, so they are constantly working on it, and if there's something specific that you're interested in trying to understand or a specific question you're trying to answer, then you can actually contact David Wharton. He's normally quite happy to field questions or develop something based on a specific question. Yeah, so that's it from me.
Thanks.